The overall goal of the following experiment is to identify the exact positions of integrating viral vectors in the genome. This is achieved by an initial linear PCR reaction with biotinylated primers to enrich for vector genome junction sequences from genomic DNA on solid phase. In a series of reactions, double-stranded DNA with known DNA sequences on both ends of the product are generated, which allows for exponential amplification of vector genome junctions. Next, sequencing adapters are incorporated to LAM-PCR products in order to sequence and characterize the unknown flanking DNA. With deep sequencing, these fragments reveal the exact positions of vector integrations. The result is a diversity of amplified junctions as viewed by gel electrophoresis.
This method provides insight into viral vector integration site distribution in clinical gene therapy patients. It can also be applied to other studies such as insertional mutagenesis screens, virus infection, cancer and stem cell clonality or T-cell diversity. The procedure will be demonstrated by Ina Kuchera, a technician from my laboratory.
For this procedure, first prepare the linker cassettes. Mix 40 microliters of each of the two LC oligos with 110 microliters of tris hydrochloride and 10 microliters of magnesium chloride in a microcentrifuge tube. Next, set the mixture to incubate on a 95 degrees Celsius heat block for 5 minutes, and then cool slowly to room temperature overnight. The next day, add 300 microliters of water to the prepared double-stranded linker DNA and filter them over a microcentrifuge tube. Spin down the filter to collect the concentrated linker DNA in the eluate. Next, recover the eluate from the filter. Add 80 microliters of water to the eluate and pipette 10 microliter aliquots into PCR tubes. Use PCR to pre-amplify the vector genome junctions in 50 microliter reaction tubes. For LAM, add between 1 nanogram and 1 microgram of sample DNA per 50 microliter reaction. For NR-LAM, use at least 100 nanograms of DNA. Do not add more than 25 microliters of template. Execute the PCR according to Table 2 in the text. And when completed, add an additional 0.5 microliters of TAC to each reaction and run the PCR program a second time. First, prepare the magnetic beads. Add 20 microliters of streptavidin-coated magnetic beads to a 1.5 milliliter tube and put them on the magnetic bead separator for 1 minute at room temperature. Then, discard the supernatant. Resuspend the beads in 40 microliters of PBS with BSA and put the beads back on the separator for another minute. Replace the supernatant with a 20 microliter wash of 3 molar lithium chloride solution. And again, put the beads on the separator for a minute. Finish by replacing the supernatant with 50 microliters of 6 molar lithium chloride solution. Now, add the magnetic bead mix to the product of the PCR reaction and allow this mixture to incubate for 2 hours to overnight at room temperature with gentle shaking. The biotinylated DNA and streptavidin-coated beads will form complexes that can be stored at 4 degrees Celsius for up to 4 days. Proceed with LAM or NR-LAM.
Due to its high sensitivity, LAM-PCR is prone to contamination if executed inattentively. Thus, a PCR-grade environment and a special attention to cleanliness are of utmost importance to successfully amplify the unknown flanking DNA without contaminating the samples.
First, expose the complexes to the separator for a minute and resuspend the DNA beads in 100 microliters of water. Next, expose the complexes to the separator for a minute and this time resuspend the DNA bead complexes in a mixture with 8.25 microliters of water, 1 microliter of hexanucleotide buffer, a quarter microliter of DNTPs, and half a microliter of clinol polymerase. Incubate this mixture at 37 degrees Celsius for an hour. Then add 90 microliters of water and expose the reaction to the separator for another minute. Then, resuspend the DNA bead complexes in a 100 microliter wash of water. Use the separator for another minute and prepare the restriction enzyme reaction after removing the supernatant. Add 8.5 microliters of water, 1 microliter of restriction enzyme buffer, and half a microliter of the restriction enzyme.
When selecting the restriction enzyme, make sure that it has no sites in or downstream of the primer binding site used in the preamplification reaction.
After allowing the enzymes to react for an hour at the ideal temperature, add 90 microliters of water. Use a separator for a minute and resuspend the bead DNA complexes in a wash of 100 microliters of water. Repeat the separation and prepare the ligation reaction. Add to the beads 5 microliters of water, 1 microliter of 10x FastLink buffer, 1 microliter of ATP, 1 microliter of FastLink DNA ligase, and a microliter of the linker cassette made at the beginning of the procedure. Allow this reaction to run for 5 minutes at room temperature. End the reaction by adding 90 microliters of water and separating the beads, followed by a wash in 100 microliters of water, and then another separation. To denature the synthesized DNA, resuspend the beads in 5 microliters of 0.1 normal sodium hydroxide and allow the mixture to shake for 5 minutes at room temperature. Then, separate out the complexes for a minute and collect the supernatant, which contains the preamplified vector genome junction. Immediately proceed with the exponential amplification, or store at -20 degrees Celsius. Begin with exposing the complexes to the separator for a minute and resuspending the DNA beads in 100 microliters of water and separate them again and remove the supernatant. For the ligation reaction, add 6.5 microliters of water, 1 microliter of Circ ligase 10x reaction buffer, half a microliter of magnesium chloride, half a microliter of ATP, 1 microliter of SS linker oligose, and half a microliter of Circ ligase. After an hour at 60 degrees Celsius, end the reaction with 90 microliters of water using the bead separator, resuspending the beads in another 100 microliters of water, using the separator again, and collecting the wash complexes in 10 microliters of water. Begin by preparing a PCR master mix as described in Table 3 of the text. Add 48 microliters of master mix to 2 microliters of LAM or NR-LAM products in 200 microliter PCR tubes and run the PCR as described in the text. As before, prepare more streptavidin beads and resuspend them in 6 molar lithium chloride. Use 20 microliters for LAM or 50 microliters for NR-LAM. Then add beads to the same volume of PCR reaction products and incubate them on a shaker at 300 RPM for 2 hours to overnight at room temperature. Collect the DNA bead complex and wash it with 100 microliters of water as previously demonstrated. Now, resuspend the complex in 0.1 normal sodium hydroxide and incubate the beads for 10 minutes at room temperature on a shaker. Then expose the beads to the separator for a minute and collect the supernatant containing the amplified DNA to a new tube. Using the amplified DNA as a template, use 2 microliters of it to run a PCR as described in Table 4 of the text. Visualize the products by gel electrophoresis. To purify the products, mix 40 microliters of them with 44 microliters of room temperature AM Pure XP magnetic beads and incubate them for 5 minutes. Then separate the beads for 2 minutes on the magnet. After removing the supernatant, wash the beads twice using 200 microliters of 70% ethanol and then resuspend the beads in 30 microliters of water. Using 40 nanograms of DNA, a fusion primer PCR can be used to add sequencing-specific adapters. Details are given in Table 5. Check the products on a gel. Viewing the results of LAM PCR by high-resolution gel electrophoresis is the best choice of diagnostic analysis. By comparison, while 2% agarose also works, it does not work as well. Clonality of NR-LAM PCR products cannot be diagnosed visually. A 2% agarose gel is, however, perfectly sufficient to determine the success of the protocol. After sequencing the PCR products, the location of the vectors can be found in the host genome. From this data, it is possible to log insertion site locations and coding regions, and close to transcription start sites.
After its development, this technique paved the way for researchers in the field of gene therapy to explore the biosafety of gene transfer vectors in both preclinical models as well as in clinical gene therapy trials.